Buenas tardes. Es para mí un placer presentar a Kelly McNagny, pero si mi jefe me dice así, yo voy a interpretar que les voy a contar una canción, ¿no? Como están en los pósters, ¿qué les parece si esperamos 30 minutos en lo que... ¿sí? Muy buenas tardes a todos. ¿Qué tal les ha parecido el foro? ¿Cómo va el foro? ¿Va bien? Sí. ¿Qué tal los pósters? Bueno, pues lo hicimos con todo el corazón y hoy tenemos un gran ponente, una gran persona y quiero presentarles, es para mí un honor, presentarles al doctor Kelly McNagny. El doctor Kelly McNagny hizo su doctorado, su PhD, en inmunología con el doctor Max Cooper. Max Cooper es ni más ni menos que el padre de la inmunología de las células B en la Universidad de Alabama en Birmingham y además de eso, después realizó sus estudios postdoctorales en Heidelberg, en Alemania. Esos estudios postdoctorales los hizo en el laboratorio de la European Molecular Biology, ¿no? Lo cual es una de las cosas más destacadas. Sin embargo, actualmente trabaja en Vancouver, en la Universidad de British Columbia, la UBC. Él está en el departamento de inmunoterapeuta y es ni más ni menos que el vicepresidente de la Sociedad Canadiense de Inmunología. Lo que ustedes tienen que saber es que ser vicepresidente significa que el próximo año la persona que se va a presentar aquí va a ser el presidente de esta sociedad, el cual es el cuerpo consultivo de Health Canada, de la Secretaría de Salud de Canadá. Así es que, sin más, quisiera recibir con un aplauso al doctor Kelly McNagny. Gracias.
Primero, muchísimas gracias a Dr. Palayo y para la invitación en este congreso muy bueno. Disculpa, pero necesito cambiar a inglés porque mi español es muy mal. Pero si tiene preguntas más tarde en inglés o en español, yo puedo responder en mi Spanglish. So, thank you very, very much for the invitation. It is a delight to be here. Can you hear? Is it okay? Okay. Every time I go to a conference, the first thing I check is the posters, and the posters are amazing here. The projects are beautiful, the work is beautiful, but most importantly, the enthusiasm for the students who are presenting has been exceptional. So I know this is a terrific conference. We could learn something from you folks. What I'm going to do today, I need a, okay. Do I need to move the slides myself, or? I'm going to tell you about a new target in cancer. This is one that we've been working on for well over 20 years now, and we're very excited about it because it's a protein and a target that we would never have undergone and discovered from a typical high-throughput screen. We stumbled onto it because we were interested in the biology, and it's led us on a very interesting trail of discovery. So I want to share some of that with you. So very first, there are a couple of things we do in my lab. The one I'm going to tell you about today is a project called Podocolixin in cancer, the CD34 family in cancer. The second thing that my lab is actually very good at, and I would be happy to talk with anybody about that later, is innate lymphoid cell biology in chronic inflammation and fibrosis. And we work on chronic lung inflammation and chronic gut inflammation particularly. So if you have an interest in that, please seek me out. I would love to collaborate with people on those projects. Okay. One quick disclosure, I have just started a company. I never thought I would start a company. I have no ambitions of getting rich from biotech, but I have learned that the only way you can get something into the clinic is to patent it and form a company around it. This company has no money, no assets. I'm trying to make this freely available, so I just want you to know that to disclosure. Okay, so for the order for today, I want to briefly introduce you to the CD34 family of sialomucins and one protein of that family called podocolixin. I'll tell you a little bit of the biology behind it and why we became interested in it. And then I'll shift to it being a marker of virtually every type of cancer as it becomes life-threatening. And we have lots of data to show that it's a particularly good marker, broad-spectrum marker of cancers that will go on to kill people. And we've also tried to develop some therapeutics to target it and prevent that from happening. So I'll share those stories with you today. In particular, I will tell you about 2 monoclonal antibodies we've developed, which seem to be doing a really good job of killing tumors in mice, and we'd like to get that into clinical trials very shortly. So briefly, this is the family portrait of the proteins we study in my lab. The first one is called CD34, which you probably have heard of. There are over 40,000 publications talking about CD34. And that's because it's the best marker of hematopoietic stem cells, the cells that you transplant in bone marrow transplantation. And to this day, it's still the best marker for purifying stem cells for transplantation. Despite those 40,000 papers, very little is known about the biology of this molecule. And many years ago, people thought, okay, either God put it on stem cells for us to purify them, or it has some kind of function. To understand the function, 2 large biotech companies knocked out the gene in mice, and the mice were completely normal and happy and healthy. And so then they became discouraged. Right about that time, we cloned a second gene called podocolixin. It's a marker of kidney podocytes, but it's also a marker of hematopoietic stem cells too. And we thought that if you knock out CD34 and there's no phenotype, maybe this molecule can compensate for those defects. And so we began to knock out that gene. At the same time, we found that there was a third member of this family called endoglycan, or podocolixin 2. This is also expressed on hematopoietic stem cells, also expressed on blood vessels. So it's a 3-gene family. Ever since we made this discovery, we've been knocking out the genes and looking at where the molecules are singly expressed for phenotypes, or making double and triple knockouts to try to understand the biology. Just to show you their common gene family, this is the genomic organization for each of the 3 genes in human. All 3 of them have 8 exons. Each exon includes a similar domain of the proteins, and they even undergo a similar pattern of alternative splicing. So it's an old family of 3 genes. Oops, if I can go back. One last thing I wanted to point out from this slide. You see here that this gene has a mucin domain, a highly glycosylated extracellular domain. So it acts as a type of a molecular Teflon, or that's what we believe. It's highly glycosylated, lots of negative charge, and it serves to repulse membranes from each other and keep cells from sticking together. We think that's the main function. So this summarizes about 20 years of my life working on these proteins. The main function we see in every case where we study these molecules is they block adhesion. If you have hematopoietic cells that express it on the surface, it keeps them from sticking to vessels. If you take it away, cells get particularly sticky and they stick to vessels. It towers above adhesion molecules on cells and prevents those adhesion molecules from working unless you clear these proteins out of the way. And we've shown that if you activate cells, very rapidly these molecules move away from other adhesion molecules and now allow cells to stick. So it's a movable Teflon. We also know that they regulate membrane topology. If you look at endothelial cells or epithelial cells and you overexpress these molecules, they expand the apical domain and they begin to sprout microvilli. So they expand apical domains and regulates apical domain size of cells. In rare cases, it can be a pro-adhesive molecule, these 3 proteins. The last thing is that we know that they are involved in chemotaxis. So each of these molecules has an ability to pair with chemokine receptors and direct directional chemotaxis into tissues. So those are the main functions of these molecules. We realize that they block adhesion makes cells more mobile and invasive. Cancer is a mobile and invasive disease. So many years ago, we began looking at cancers to see if any of these molecules are upregulated on tumor cells. And the first place we looked was in breast cancer. So this is a study where we took 300 breast cancer samples that we got from the British Columbia Cancer Center. We stained them for all three of the molecules. Podocolixin was the only one on breast cancer, and it was on about 5 to 10% of the cases. The good thing about these samples was that they were over 20 years old, and so we could follow up what happened to the patients if they had podocolixin on their tumor at diagnosis or not. And what we found was that if you look at the population as a whole, 70% of those patients were alive 20 years later. And that's because if you catch breast cancer early and you remove the tumor and you have no lymph node invasions, usually you go on to have a happy, healthy life without cancer. On the other hand, if you looked at the patients that had podocolixin on their tumor at diagnosis, every single one of them was dead within 15 years. And it turns out that if you look at the odds ratio, podocolixin expression by your tumor at diagnosis makes you 8-fold more likely to die from that tumor than if you don't have it. So it's a good predictor of bad disease. And it's an independent predictor. So if you try to compare podocolixin expression with tumor size or tumor grade or expression of loss of expression of P53 or HER2, it's an independent marker, and it's a much better marker of bad outcome disease. So that got us excited. We've looked at many other tumors, and so have other groups. I'm just going to give you one more example. This was a study done in Sweden by a group that looked at over 500 colorectal tumors. And once again, you see that if you had low podocolixin on your tumor or medium expression of podocolixin on your tumor, you did well. If you had very high expression, you did very poorly. But more interestingly, this was a study where they were looking to see whether surgical removal of the tumors alone or surgical removal plus chemotherapy gave you a better outcome. So we were lucky with that, and we binned the samples that were podocolixin positive and did or did not have chemotherapy. Turns out that if you have high levels of podocolixin and no chemotherapy, again, you do very poorly. But if you had high levels of podocolixin and you got adjuvant therapy, you did just as well as people who had no podocolixin on their tumor at all. So what that tells you is it's a good theranostic. You can use this molecule to determine whether you should or should not get chemotherapy, and that's a big deal in colorectal cancer because not everybody wants to go through chemotherapy. It's a tough therapy. If you know that you need to do it, you should. If you don't, you might decide not to have it. Okay. There have been many other studies looking now at podocolixin in tumors, and what's remarkable to me again is that it's on virtually every subclass of tumor as they become poor outcome. Here are the list of tumors in men and in women according to the amount of death associated with those particular tumors in the US. Everywhere you see an arrow, its podocolixin expression has been analyzed and it turns out to be a marker of bad disease. Everywhere you don't see an arrow means that they haven't looked yet. So we think that this is a good marker of all tumors, and in honesty, I think that it should be used to test everybody's cancer and determine whether you should get aggressive therapy or not. And for the life of me, I can't understand why that hasn't happened yet. I think some of it is a bit of politics, and I think a lot of it is corporate greed, but we can talk about that later if you like. Okay. So that shows you we've got a good marker of disease. We should be using it in the clinic, but maybe a more important question is it a target for therapy? Can we use this knowledge in order to treat cancer? So that's something we've been working on very hard for the past few years. Is it involved in the tumor progression process? Here's an example where we showed that it is. So here we've taken a very aggressive breast cancer cell line called MDA231. It's a triple negative tumor. It forms great tumors when you xenograft it into mice. That cell line expresses very high levels of podocolixin, and so we knocked it down in one group of cells and labeled them red. We knocked put a control vector in the other group of cells and labeled them green, mixed them together, put them in mice, and then asked 6 weeks later, which of those cell types gave you tumors? Did they both give tumors, or did podocalyxin knockout have an effect? And if you do that experiment, you get a really interesting result. So here are the lungs of these mice where we've treated them with the tumors. And what you see is if we knocked out podocalyxin in the green cells, all of the tumors came from red cells. So knocking out podocalyxin crippled the ability of those tumors to form. Only the cells that have podocalyxin could form tumors. We flipped the labels around and did the same assay. Here you see if you knock it out in the red cells, you get no tumors from the red cells and only get tumors from the green cells. So in this intravenous assay, knocking out podocalyxin cripples the ability of tumors to metastasize to lung. We've also taken the tumors and injected them in the flanks of mice. If you don't knock out podocalyxin, you use the wild type cells. This is the muscle layer in pink. These are the tumor cells in purple. You see they shred right through the muscle and they begin burrowing into the bone, just like breast cancer does. If you knock out podocalyxin, the tumors grow incredibly slow and they tend to stay encapsulated and they don't invade the muscle. So we've crippled the abilities of these to metastasize and invade tissue as well. One other detail. It's well known that if you look at tumors, there's a subset of the tumor cells that have an ability to metastasize. It's not all of the cells, it's a subset. And people call these tumor stem cells. There's an assay for tumor stem cells in vitro. You can take out tumor cells, put them in suspension culture, and look for formation of tumor spheres. We've done that with the podocalyxin knockdowns, either in red or in green. In both cases, we've knocked down the ability of tumor spheres to form by either 3-fold or 10-fold simply by getting rid of that one gene. So we think that this is having an effect on tumor stem cell behavior, the ability of tumors to move and metastasize to distal tissues. That's breast cancer. We've now made knockout cell lines in a variety of different cancers. Here's ovarian cancer cell line that we've grown in mice. Wild-type cells with podocalyxin form tumors, ones without grow very slowly. We've done the same thing with a pancreatic cell line where we get good growth with podocalyxin. You knock it out, it's very difficult to get tumors to grow at all. So it seems to be true of every cancer we've looked at. Now, knowing all of that, because if you can get rid of that protein, you've got a great way of treating cancer. So we thought, is there a way we can target the protein? And so we got together with the Center for Drug Research and Development at British Columbia, and we made over 200 monoclonal antibodies against podocalyxin, hoping that one or another would be able to suppress tumor growth. We screened 28 million B cells. We came up with 200 monoclonals. Then we had to do a counter screen, and this was a very painful screen, I can tell you, because if you knock out podocalyxin on the cells, they don't grow in mice, but if you just look at them in a dish, they don't look like they changed their phenotype very much. They still divide in tissue culture. They look the same way if you get rid of podocalyxin. So really the only assay we had was to stick tumors in mice and see if we could cure them. And so that's what we did. We took tumor cells, coated them with monoclonals, put them into mice, and asked if any of the antibodies we had developed slowed down tumor growth. Using that screen, we came up with 2 monoclonal antibodies. One is called PODO447. This one doesn't slow tumor growth, but I'll show you later that it is exquisitely specific for tumors and doesn't touch any normal tissues. The second antibody we have is called PODO83. This binds to a different domain of podocalyxin, and it really blocks tumor growth, as I'll show you now. So here's that screen I told you about. We coat cells, we put them in Matrigel, we inject the Matrigel in the flank of the mice, and we wait 2 weeks to see if any of the tumors grow more rapidly or more slowly. And with 9 of the antibodies we tried in this assay, the tumors grew very rapidly, but this one antibody completely blocked tumor growth. So that looked good. Normally you don't know you're going to get cancer before you have cancer, so not very useful. So we tried a different assay. Instead, we waited until the tumors were about 100 millimeters cubed in the mice. This is about the size of a lentil bean. And then we began treating with the antibody in 4 doses, or a control antibody. Control antibody, the tumors grew. Our antibody, no growth at all. So as a naked antibody, this seems to be a good blocker of tumor growth. Here's what the tumors look like, and this is the size of the tumors. Okay, that's pretty good. That's a good-sized tumor on a mouse. We've been able to block it. We really pushed it as far as we could, so now we waited until the tumors were about 1 centimeter cubed, about the size of a hazelnut. So we waited until they're 1 centimeter cubed, then we began treating with antibodies, and if you're optimistic, you can say our antibodies seemed to slow the growth a little bit. At this point, we had to kill the mice because the tumors were getting too large. But one thing we know about this particular tumor cell line is right about this stage, you start to get metastases from the primary site to the lung. And so we had a look at the lungs of the same mice. And if you look there, it's really night and day. So here are the lungs of mice that got a control antibody. You can see that the lungs are absolutely riddled with tumors. There's too many tumor nodules to count. And here are the mice that we gave our antibody to. And you can see that you have a few very tiny tumors on the lung, but nothing like these mice. So not only are we slowing the growth of primary tumors, but we're blocking metastases to peripheral and distal sites. Okay, what's special about this antibody? So I told you, we made 200 monoclonal antibodies against podocolixin, but only this one shows this behavior. Now, what we know about this antibody is it binds right to the juxtamembrane site, right next to the plasma membrane. And knowing the scale of the podocolixin protein, an antibody would be almost the same size as this entire protein. So it's really going to block this site. So if there were other proteins associating with podocolixin, we think that antibody would block them. And one thing interesting to know about podocolixin is it binds a protein called NRF1. This is a scaffolding protein that binds to the C-terminal of podocolixin, but it also has a binding site for the C-terminus of other proteins. And this is a short list of all the other proteins that associate with NRF1. So you have tyrosine kinase type receptors like PDGF receptor, VEGF receptor. You have transcription factors like beta-catenin and some of the Hox genes. You have lipid kinases and a whole slew of G-protein coupled receptors, all of which bind to this protein. And we know that podocolixin recruits this protein to apical domains, as I'll show you in this slide. So here's NRF1 expression in a normal epithelial cell line in green, and you can see it's in the cytoplasm of this monolayer. Here's podocolixin expression when we overexpress it in that same cell line. When you overexpress podocolixin, now all the NRF moves up to the apical membrane. So it's recruiting NRF and all of those proteins that are associated with it to a new domain in the cell, and we think that maybe our antibody is blocking that association. That's something we're investigating now. So we think that it regulates signal transduction, and we're blocking that with that particular antibody. Okay, quick summary up to this point. Podocolixin is a marker of poor outcome in every tumor that we've looked at, and I think it's probably true for just about any tumor. Loss of podocolixin cripples the ability of tumors to grow in mice and likely in human as well. Podo83 antibody binds the core protein peptide and appears to be blocking function. It has a high specificity for human podocolixin and not mouse podocolixin, and that's something I want to emphasize because it does present a problem. That antibody, podocolixin, is normally expressed in hematopoietic stem cells, kidney podocytes, and vascular endothelia. And I can't guarantee you that if we use this antibody in patients, we wouldn't damage those tissues. We have reason to think we wouldn't, but I can't guarantee it. So, we've tried to address that now by making a transgenic mouse. And what we've done is we've taken the human gene, shown here, and we've replaced the mouse exons with the human exons that encode the binding site for that podocolixin antibody. So we've made a humanized mouse. Here is a kidney where we see normal podocolixin expression of the mouse podocolixin. It doesn't bind the human antibody. Here we've made our transgenic mouse where we put the human protein in. It's lost binding of the mouse antibody, but now binds very potently the human antibody. So now this mouse expresses human podocolixin in all the right tissues, and we can check for toxicity. We've injected this mouse with milligram amounts of antibody and not seen a single defect in those mice. So it looks like whatever this antibody is blocking in terms of function, it's selectively affecting tumor growth, but not normal tissue growth. And we're very excited about that because it suggests that a clinical trial with this antibody might go really, really well. Okay. So, enough about podocolixin 83. Good target, good antibody, might be useful as a naked antibody. But I want to talk now about a second antibody we made. And this one came out of a second screen. So, knowing that human podocolixin was expressed on normal tissue, we screened for antibodies that would bind to the tumor glycoform of podocolixin, but not the normal form. And we knew that tumors glycosylate the protein differently. And out of those 200 monoclonal antibodies, we came with one that really binds human podocolixin on tumors, but not on normal tissue. And I'll show you that now. So, here are FACS profiles showing the binding of the antibody. Here are HUVEC cells, normal human umbilical cord blood cells. They bind podocolixin, but they don't bind our antibody that's glycosylation specific. They don't bind the 447 antibody. Here's a brain tumor, A172. It's a glioblastoma. Here's a breast cancer cell line. Here's the pancreatic cell line. All of them express the podocolixin core protein and the tumor glycosylation. Okay? So this antibody appears to be tumor specific. We know that the glycosylation it recognizes is tumor specific and podocolixin specific. And the reason we know that is if you knock out podocolixin in each one of these cell lines, you of course lose binding of the core antibody, but you also lose binding of the glycosylation specific antibody. So it's not just recognizing the glycosylation, it's recognizing the glycosylation plus the podocolixin protein. This shows you it in histology. Here's a normal kidney stained with the podocolixin core antibody. This is our tumor-specific antibody. It doesn't stain kidney, doesn't stain endothelia or any other cell type in any other tissue, and we've looked at every tissue we could lay our hands on. So again, it looks like it is exquisitely tumor-specific, a very unusual antibody. Now, what is it recognizing in terms of glycosylation? So we know that human umbilical cord blood cell lines, or human kidney cell lines, HEK293, express podocolixin, and they express that glycosylation. We got together with a group in Denmark that had a library of these cell lines. In each of these different subclones of the cell lines, they've knocked out glycosylation enzymes that change the O-link glycosylation on the cells. If you knock down terminal glycosylation, our antibody still binds. If you trim it down a bit further, our antibody still binds. If you cut back to the very shortest glycosylation, our antibody actually binds even better. So it binds an underglycosylated protein. But if you cut back one more sugar, you lose all binding. So our antibody recognizes what's known as a T-antigen glycosylation. It's a tumor-type glycosylation, but only in the context of podocolixin. Okay, now, one other interesting thing. We have some cell lines that express uniformly podocolixin at very high levels. They express the core protein at high levels, but that are heterogeneous with expression of the tumor glycosylation. So you look at the cell line, 100% positive for podocolixin, but only some of the cells express the glycoepitope and some cells don't. And that's shown here. If we sort them, we sort the bright and we sort the dim, they look like this. We then went on and looked at gene expression in those sublines of the cell line. And if you look at gene expression, you find that in the cells that lack the glycosylation epitope, they all have normal epithelial gene expression. If you look at the cells that have that tumor-specific glycosylation, they express a variety of genes associated with epithelial to mesenchymal transition. So they upregulate the snail and slug proteins, they upregulate ZEB1, they change the expression of a variety of adhesion molecules. These are all genes linked to EMT. So we think that within this cell line, our antibody picks out the tumor stem cell subset of that cell line. And we think that somehow, for some reason, changes in glycosylation are linked to that phenotype intimately. Okay, now that antibody recognizes some tumors, not as many as the POTO83 antibody. Here we've looked at ovarian cancer. And if you look at the high-grade serous ovarian cancer, you find that 97% of them are positive for potocolixin, and that's because you diagnose ovarian cancer very late usually. It's grown for quite a bit before you actually identify it. All of those tumors are positive for potocolixin. 62% are also positive for that tumor-specific glycosylation. So we think that you could use that tumor glycoantibody for treating a very wide variety of ovarian cancers. Same is true for uroepithelial tumors, bladder cancer tumors, pancreatic tumors are a little bit lower, but still quite good. And a variety of breast tumors, it looks like now about 28% of breast tumors with our high affinity antibodies are recognized by our antibody. Okay, ovarian cancer. Here's what the staining looks like. Every tumor expresses the potocolixin core protein strongly. The expression of the 447 glycosylation antibody is variable. Some are completely negative, some are weak, and some are fairly strong. What's different about these different subtypes of tumors? One thing interesting we noticed was that if you look at the level of glycosylation-specific antibody staining versus the number of B cells or T cells, CD4 or CD8s, in the stroma or the tumor, you see this really interesting inverse correlation. The more T cells you have, the lower the 447 staining. The more B cells you have, the lower 447 staining. The more 447 staining, the lower immune infiltrates. What we call this is immunologically cold tumors, tumors that have no immune response. These are always the worst of the worst tumors. They're the ones that progress unabated and are not cured by the patient's immune response. This is a better marker for those cold tumors than any other marker out there, and I'm just showing you here CA125. This is a commonly used marker for ovarian cancer. It tells you whether you have an ovarian cancer or a benign tumor. You can see that there's no correlation between the 447 epitope and that marker. In fact, it looks like CA125 recognizes the hot tumors, not the cold ones. Mesothelin, as well as folate receptor, also don't distinguish as well as our 447. Our 447 antibody is much more specific. All by itself, that antibody does nothing to tumors, but we've now coupled it to a toxin, MMAE. This is a aromatase inhibitor. If you couple that antibody to a toxin, it's very effective at killing pancreatic cancers, pretty effective at killing ovarian cancer tumors. And we know it's specific because if we knock out potocolixin, then we don't get any killing of the tumors. So it looks like when you couple it to an antibody, it's an effective tumor-killing agent. Here we've treated mice with pancreatic cancer with the antibody at 2 different doses, 4 mg per kilogram or 2 mg per kilogram, 2 different experiments. You can see that if you got the potocolixin antibody, the mice were well cured from the tumors. Without it, they weren't. The tumors grew on unabated. One last couple of points, and then I'll finish up. So here we're looking at treatment of NSG mice. These are mice with no T cells and B cells or NK cells. Very easy to transplant tumors in those mice because they have no immune response. And we're comparing it to treatment of nude mice, which have no T cells, but still have NK cells and B cells. How effective is our antibody in those 2 scenarios? If we treat the NSG mice, we slow down the tumor growth. All these ones in blue are tumors that were growing with a control antibody. In red are tumors that were growing with our potocolixin-specific antibody. You can see that we slowed the tumor growth with these early treatments here, Eventually, the tumors came back, but they grew very, very slowly. With the nude mice, we actually cured most of them. And what I think that's telling you is that if you can kill the tumor stem cells and you can slow down the tumor growth, the residual immune response will clear the rest of the tumor cells that's left over in the patient. And that's a really important thing because this is more closely mimicking what you have in a patient than the NSG mice, I would argue. Okay? One last detail about this experiment, and we're going to focus now on these mice. So remember, if you got a control antibody, the mice had to be killed early. If you got our antibody, they had to be killed late because they had slow-growing tumors. If we compare those tumors, here's what you see. So these are mice that were treated with a control antibody. They're very positive with the POTO83, so they still express potocolixin, and they still express our glycoepitope. Okay. Here are mice that were treated with our glycospecific antibody. And what you can see is if you look at POTO447 expression, there's a little bit of it still in the very center of the tumors, but most of the cells have turned it off. Okay? And remember, these are tumors that we harvested a month later, so they're growing very slowly. There's a few cells that are still positive. Now, I assumed that because this antibody recognizes a glycosylation, well, the tumors are just going to change the glycosylation and they're going to escape and they're going to grow and that's going to be the end of our therapy. It's not true. What happens is they turn off expression of the core protein. So this is the core protein expression. You can see the only cells that are still expressing the antigen are in the very center of the tumors. These cells can't escape by changing glycosylation. The only way they can escape is by turning off the core protein. And I've already shown you in 3 different examples where turning off the core protein slows tumor growth to where it's treatable. So we think this antibody is going to be a great therapy, and we don't have any worry about toxicity because it's not expressed on any normal tissues. One last detail. Antibodies are now being converted into single-chain antibodies for therapy. They're also being converted into CAR T-cell therapy, where you put a single chain on a CAR receptor and transfect it into a T-cell for these very expensive therapies that they have access to in the States. We're trying to build that in Canada very slowly. We don't have as much money, but we're trying. We've converted the 447 antibody into a CAR. Here I'm expressing that CAR in human T cells, CD4 and CD8 cells. You can see that it's well expressed. If we take those control CARs, or we take our POTO-CARs, and we mix them together with this glioblastoma cell line, A172, control CAR T cells don't do much of anything to the tumor. You get about 10% killing, but nothing more. With our POTO-specific CARs, we get complete killing if we increase the dose. So it looks like as a CAR T therapy, this would work as well. And again, I'm reminding you that glycosylation epitope isn't on any normal tissue, so it should be a well-tolerated therapy. Okay, so quick summary. 447 detects a highly, exquisitely specific glycosylation epitope on potocolixin. It's a T antigen type epitope. Its expression is linked to an EMT gene signature. It's maintained on ovarian cancers post-chemotherapy. I haven't shown you that, but we've looked at ovarian cancers that have undergone chemotherapy, and they still express that glycosylation. So they would be good candidates for now being treated with this antibody. Its expression inversely correlates with lymphocyte infiltration, so it marks cold tumors, and we're checking to see if that happens in other tumors too. It's more specific for cold tumors than any other antigen out there, and there's no evidence that tumors can escape by changing the glycosylation. One thing to think about, we screened 28 million B cells to find that one antibody, okay? We got very lucky. But now that we know that there's a subset of tumors that glycosylate tumors differently, you could now go back and sort those cells and pull out any of your favorite tumor antigens that are glycosylated, MUC1, any of the other mucin-type proteins, and try to make an antibody that recognizes the alternate glycosylation. And now maybe instead of having this one antibody, you could have a whole panel of antibodies that are exquisitely tumor-specific and don't recognize normal proteins. And that's really the holy grail for cancer therapy. Something that's tumor-specific but not going to damage normal tissue. Okay, last general summary, Podoclixin blocks function. It's a function blocking epitope, still excited about that one. 447 is exquisitely tumor specific. It functions as an ADC to kill tumors. Residual tumors after treatment seem to grow slowly and are mopped up by the endogenous immune response, and it works as a CAR. This is my team, some great people. These are Cal Ross-Kelly has been my long-term collaborator in cancer. I knew very little about cancer before I started working with him. He's a terrific guy. This is his postdoc, Pam. And my postdoc, Michael Hughes, has been with me for over 15 years. He just recently left. I'm missing him already, but he's instrumental in everything we've done with Podoclixin. This is the rest of my team. It is an incredibly diverse group, as you can see. We love diversity. In fact, it's our lab motto. Powered by science, strengthened by diversity. Everybody who comes from my lab comes from somewhere else. They come from Europe, they come from Asia, they come from the Middle East, they come from South America, they come from Latin America. Those different viewpoints and ideas have been instrumental in making my lab a better place. And I think it's really important to say that, especially this day and age when we've got such crazy things happening in the US. So I'll leave you with that. Thank you.
Pues muchísimas gracias. Very thank you for this talk. Y le otorgamos un reconocimiento por parte de la Coordinación y un regalo, como ya lo habéis mencionado. Muchas felicidades. Muchas gracias por venir.
Gracias. Finalmente, si hay preguntas, por favor, pregúntame, después o ahora.